In the last episode, we defined the positive controls in life science experiments. In this episode and the next, we will examine the positive controls that the Nobel Laureates used in our Milestone Article 1. In two comments near the end, we will also talk about how carefully the laureates named the RNAs and how carefully the laureates designed an experiment in relation to positive controls. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto, learn the skills, enjoy academic life. Last episode. In the last episode, we started discussing a new substep in relation to figures in our Milestone Article 1. It was Step 11-6, Identify the positive controls of experiments. To start the discussion, we defined the positive controls. Then we refined that definition so that it will encourage us to think practically and cautiously about positive controls. The modified definition was, a positive control is a sample where the expected outcome is thought to be present. The expression in the original definition, known to be present, is now replaced by the key expression, thought to be present. We wanted to modify the definition of positive controls because a positive control in one experiment does not necessarily work as a positive control in another experiment. We will talk about one example in the next episode. Today's episode. We are still in the step 11-6 about positive controls. After defining the positive controls in the last episode, we will see the examples of positive controls in 3 figures of our Milestone Article 1. Today, we will focus on 2 of them. In the next episode, we will focus on the third figure, which is our milestone graph, figure 3D, D as in dog. Positive controls in our Milestone Article 1. Let's look at the examples of positive controls. They are in figures 1 and 2b, B as in boy. These are 2 and 1 figures before our milestone graph, which is figure 3D. In episode 16, we talked about the beautiful vertical or category axis labels in our Milestone Article 1. At the time, we discussed figure 2b before talking about figure 3d because it showed a more refined structure of axis labels than our milestone graph. After figure 2b, we discussed figure 3d. We found the sequential discussion to be very efficient. Let's do the same here. Let's start with earlier figures, figures 1 and 2b, and then let's discuss our milestone graph because it will be easier for us to understand it after those earlier figures. 2 major questions of the Milestone Article 1. Before going into details of positive controls, it will be helpful for us to briefly talk about what the laureates did in the experiments of figures 1 and 2b. In figure 1, the laureates had shown that when RNAs were experimentally introduced into immune cells called the dendritic cells, the inflammatory cytokines were released from those cells. So RNAs were shown to stimulate the cytokine release. Among the RNAs, the especially strong stimulator was the RNA called in vitro transcribed RNA. In vitro means outside the living body and in an artificial environment, often in a test tube. And the transcribed means that an RNA was synthesized from a DNA template. The in vitro transcribed RNA was generated in the lab from a DNA template using the 4 basic ribonucleotides, ATP, CTP, GTP, and UTP. It was named the unmodified in vitro transcribed RNA. It encoded a biological protein, specifically a protein called luciferase of fireflies. Because the unmodified in vitro transcribed RNA was one of the strongest stimulators of immune cells, such RNA can be considered as a positive control for the immune cells. The laureates also found that naturally occurring biological RNAs were variable in terms of how strongly they stimulated the cytokine release. It appeared to them that the RNAs with more unmodified nucleosides tended to stimulate the cytokine release, whereas the RNAs with more modified nucleosides did not. But there was no proof yet. Based on these results of Figure 1, the laureates obtained two major questions of the Milestone Article 1. The first question was, when the cytokine release is stimulated by the unmodified in vitro transcribed RNAs, what mechanism do the RNAs activate? More specifically, which subtypes of receptors for RNA, called the toll-like receptors, do the RNAs activate? And the second question was, do modified nucleosides of RNAs suppress the RNA's ability to stimulate the cytokine release in comparison to the unmodified nucleosides? They answered these two questions mainly by the experiments in Figures 2b and 3d. For these experiments, the laureates used the in vitro transcribed RNAs because they could manage the ratio of modified versus unmodified nucleosides in the RNAs. This was done by changing the ratio of modified versus unmodified nucleotides in the reaction solutions during the RNA synthesis in the laboratory. Experiments in Figure 2b of Milestone Article 1. Here are 4 features about the experiments in Figure 2b, one figure before the milestone graph. The experiment was designed to address the two questions that we just talked about. 1. They prepared a model cell line, the human embryonic kidney 293 cells, that we often call HEK293 cells. The laureates used these cells because genetic engineering is easy with those cells. 2. From the cell line, the laureates prepared 4 different types of cells that each expressed a different subtype of toll-like receptors. Those cells expressed either toll-like receptor 3, toll-like receptor 7, toll-like receptor 8, or none of the toll-like receptors. 3. They introduced RNAs or related synthetic compounds into these cells. And 4. The laureates measured the amounts of the released cytokine interleukin-8, abbreviated as IL-8, and they compared the released amounts among different conditions. So in summary, the laureates tested the effects of multiple types of RNAs and related compounds on the cells that expressed different subtypes of toll-like receptors. How Figure 2b looks. Here I will describe Figure 2b a little bit so that you can visualize it better. Figure 2b looks like a table. But instead of showing a number, each entry in a table cell, not a biological cell, shows a horizontal bar. And the bar length represents the amount of released cytokines under the specific condition for the table cell. A table has rows and columns. In Figure 2b, the rows represent experimental categories. Specifically, they indicate what RNA or related compound was introduced into the HEK293 cells. And the columns represent the types of cells that expressed different toll-like receptors. There are 4 columns. They correspond to cells with toll-like receptor 3, 7, 8, or no toll-like receptors. Now that we have understood the basic features of the experiment in Figure 2b, let's talk about the positive controls in the figure. 4 positive controls in Figure 2b of Milestone Article 1. There are 4 positive controls. They were used beautifully in this figure. 2 positive controls were synthetic compounds called poly-IC and R848. And 2 other positive controls were unmodified forms of in vitro transcribed RNAs called RNA 1571 and RNA 730. Let's talk about each of them. Positive control in Figure 2b, one synthetic compound, poly-IC. One positive control was a synthetic chemical, poly-IC. The abbreviation stands for polyinosinic polycistidylic acid. It is a synthetic analog of double-stranded RNA, which is present in some viruses. One strand is made up of inosines, and the other strand is made up of cytidines. Poly-IC was used as a positive control for stimulating the toll-like receptor 3. The results in Figure 2b were consistent with this statement. I will tell you 3 results. Poly-IC induced strong interleukin-8 cytokine release in HEK293 cells expressing toll-like receptor 3. But poly-IC did not induce cytokine release in cells expressing toll-like receptors 7 or 8. Poly-IC did not induce cytokine release either in cells expressing no toll-like receptors, labeled as vector. The cells that did not express toll-like receptors could serve as a negative control for poly-IC. So these results indicate that poly-IC activates toll-like receptor 3 specifically without activating toll-like receptor 7 or 8, or without stimulating the cells randomly. Positive control in Figure 2b, another synthetic compound, R848. Another positive control in Figure 2b was a synthetic small chemical, R848. R848 was used as a positive control for stimulating the toll-like receptors 7 and 8. The results in Figure 2b were consistent with this statement. I will tell you 3 results. R848 induced strong interleukin-8 cytokine release in HEK293 cells expressing toll-like receptors 7 or 8. But R848 did not induce cytokine release in cells expressing toll-like receptor 3. R848 did not induce cytokine release either in cells expressing no toll-like receptors. The cells that did not express toll-like receptors could serve as a negative control for R848. So these results indicate that R848 activates toll-like receptors 7 and 8 specifically without activating toll-like receptor 3, or without stimulating the cells randomly. If you look at Figure 2b, you will find interesting behaviors of the bars. As we discussed, the length of a bar represents the amount of released cytokines, interleukin-8, from the stimulated cells. The lengths of the bars for poly-IC and R848 were complementary to each other. I will tell you 3 features here. Poly-IC bar was long for toll-like receptor 3 expressing cells, but short for toll-like receptors 7 and 8 cells. Conversely, R848 bar was short for toll-like receptor 3 expressing cells, but long for toll-like receptors 7 and 8 cells. And the common behavior between poly-IC and R848 was that both their bars were short for cells that do not express toll-like receptors. So these 2 positive control compounds, poly-IC and R848, can be used to indicate which subtypes of toll-like receptors are expressed in the cells that the experimenters use. Positive controls in Figure 2b, 2 RNAs. 2 RNAs were also used as positive controls. The RNAs were RNA 1571 and RNA 730, and both were in vitro transcribed RNAs. So the laureates could prepare the unmodified and modified forms of these RNAs. As we discussed in episode 15, the number in the RNA's name indicates the number of nucleotides. So for example, the RNA 1571 is a specific RNA with 1,571 nucleotides. Here are the 2 main results in Figure 2b. The unmodified forms of these two RNAs showed long bars in all cells expressing either of the three toll-like receptors, toll-like receptor 3, 7, or 8. In contrast, these RNAs showed short bars in cells expressing no toll-like receptors. And we can also notice that the lengths of the bars were similar to those of the positive controls. That is, long bars of RNAs were like the long bars of poly-IC in cells expressing toll-like receptor 3. Long bars of RNAs were like the long bars of R848 in cells expressing toll-like receptors 7 or 8, and short bars of RNAs were like the short bars of poly-IC or R848 in cells expressing no toll-like receptors. So these results in figure 2b indicate at least three things for us. 1. Unmodified forms of RNAs activate all those toll-like receptors equally. 2. The unmodified forms of RNAs do not randomly stimulate the cells expressing no toll-like receptors. And 3. The unmodified forms of RNAs can serve as positive controls for stimulating the cytokine release from HEK293 cells by activating all those toll-like receptors equally. Please recall, Figure 1 showed that the unmodified forms of RNA can serve as a positive control for stimulating the cytokine release from immune cells. This third point of Figure 2b made the idea of Figure 1 more clearly by talking about the activation of three subtypes of toll-like receptors. I now have two comments. Comment 1. How carefully the laureates named the RNAs. This is a comment about how the laureates named the specific in vitro transcribed RNAs and how the naming rule has evolved through multiple papers. We already discussed that the number in the RNA's name indicates the number of nucleotides. But the naming rule was different initially. I'm going to show you two papers that Dr. Karikó, one of the laureates, published before the Milestone Article 1. In 1998, Dr. Karikó published a paper. It was published 7 years before the Milestone Article 1. In the paper, Dr. Karikó used only one RNA. This was RNA 1866 that was named and used in the Milestone Article 1. But how was it called in the 1998 paper? It was called simply as RNA or messenger RNA. It was a very general name. It was only occasionally called by a very technical name, mTev-LAC-A50, based on the name of the DNA template. By the way, the authors used cultured cells for the experiments in the 1998 paper, and the culture medium contained an animal serum. This reminds us of our story about negative controls. There, the animal serum played an important role in characterizing the culture media. In 2004, Dr. Karikó and Dr. Weissman published a paper together. It was 1 year before the Milestone Article 1 was published. In the paper, the laureates used 4 messenger RNAs. They were named messenger RNA 1, messenger RNA 2, messenger RNA 3. 2 of them, messenger RNA 1 and 3, corresponded to RNA 1866 and RNA 1571 in the Milestone Article 1, respectively. Messenger RNAs 1 and 3 sound very broad and vague. In 2005, the laureates published the Milestone Article 1. They used 5 in vitro transcribed RNAs. In the order of decreasing lengths, they were named RNA 1866, RNA 1571, RNA 730, RNA 713, and RNA 497. So if we focus on one example, the name changed this way. RNA or messenger RNA in 1998 became messenger RNA 1 in 2004, and then it became RNA 1866 in 2005. But These naming conventions were reasonable in each paper. RNA or messenger RNA in 1998 was a reasonable name because only one RNA was used in the paper. Messenger RNAs 1, 2, and 3 in 2004 were reasonable names because multiple RNAs were used in the paper and they had to be named differently. RNA 1866, RNA 1571, and so on in 2005 were reasonable names because, again, multiple RNAs were used in the paper and the names were specific about the lengths. They made sense because a part of the Milestone Article 1 dealt with the relationship between the degree of suppressing cytokine release and the number of modified nucleosides in an RNA, and therefore the percentage occupation in an RNA. And this was shown in Figure 5. The authors used RNA 1571 there. The number of nucleotides in an RNA is critical information for calculating the percentage occupation of modified nucleosides. So, the naming rule evolves even for the same RNAs, depending on the contents and contexts of the papers. Comment 2. How carefully the laureates designed an experiment. Today we talked about the experiment in Figure 2b. The laureates were more careful than how I described it. In the laureates' 2004 paper, they had found that the parental HEK293 cells express a low level of endogenous toll-like receptor 3. This situation was fine when the cells were engineered to express a large amount of toll-like receptor 3 in Figure 2b. But this situation was not appropriate for the three types of cells that were engineered to express toll-like receptor 7, toll-like receptor 8, or no toll-like receptors. This was because if RNA stimulates the cytokine release from the HEK293 cells, the laureates would not be able to tell whether the RNA activated toll-like receptor 3, which was present in any cell, or toll-like receptor 7 or toll-like receptor 8, which were expressed additionally. In these three types, the endogenous toll-like receptor 3 was removed by a method called a gene silencing or gene knockdown using small interfering RNAs. And then additionally, either toll-like receptor 7, toll-like receptor 8, or no toll-like receptor was expressed. In Figure 2b, those cells lacking the endogenous toll-like receptor 3 were labeled by asterisks next to the expressed toll-like receptor subtypes. For example, toll-like receptor 7 asterisk means that the cells express toll-like receptor 7 in the absence of endogenous toll-like receptor 3. This is another example of helpful labels in the laureates graphs. This was similar to the category axis labels of the same graph that we discussed in episode 16. So we can clearly see that the laureates had designed an experiment and designed the figures very carefully. A careful design of experiments leads to a solid result. This is the end of the main content today. Let's wrap up. Summary. Today we have reviewed the experiments in Figures 1 and 2b of our Milestone Article 1, and we checked the positive controls there. They were used to address two major questions of the Milestone Article 1. The first question was, when the cytokine release is stimulated by the unmodified in vitro transcribed RNAs, which subtypes of toll-like receptors are activated? And the second question was, do modified nucleosides of RNA suppress the RNA's ability to stimulate the cytokine release in comparison to the unmodified nucleosides? To address these questions, the laureates used 4 positive controls in Figure 2b. 2 positive controls were synthetic compounds, poly-IC and R848, and 2 other positive controls were unmodified forms of in vitro transcribed RNAs, RNA 1571 and RNA 730. In the next episode, we will continue to talk about the positive controls. we will focus on the positive controls in our milestone graph, figure 3D. Your action. If you like my podcast, please share it with your friends, colleagues, mentors, or anybody who would be interested in it. This way, you and I can extend the reach. The bigger the audience becomes, the more feedback we can get, the better the episode quality will grow, and the more you and I can learn. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.